right, so today we'll be talking about the amygdala, or as I've written above, the amygdaloid complex, and if it's plural, the amygdalae, as such. And I'll be talking about these 6 areas. First, functions. Even though primarily we're going to be talking about the anatomy of the amygdala, it's good to have a basic understanding of the functions, and I'll cover them more in depth in a follow-up video. Secondly, the location of the amygdala. Thirdly, blood supply. Fourthly, and probably most importantly, the regions. Fifth, a basic neuron count, giving an idea of how many neurons are located in there, and the afferent pathways. Alright, so let's talk about the functions. And these are just a list of basic functions. So first we have fear and aggression. This is for basic conditioning experiments that you might have seen. Emotion, reward, and the emotion is both positive and negative emotions, which becomes pretty important when looking at lateralization between the amygdalae. Autonomic responses, for example, to stress, and memory. Okay, so these are just a basic idea of the functions that are present in the amygdala. The location of the amygdala. Now, I will be linking a video below of precisely the location with a post-mortem dissection. It's a very good video that gives you an idea of limbic structures and how they look in relation to other brain regions, but I'll just quickly draw out an idea of where it is. Alright, and so we'll have the temporal lobe, and we'll be talking about the right-sided amygdala for the purpose of this video when we talk about the regions and I show the cytoarchitectonic map. But for this depiction from the front, you see it's located on the frontal medial part of the temporal lobe. And if we draw it from the side, again, it will look something like this. And the amygdala will be stationed right in there, right in this region. And so, yes, frontal medial temporal lobe. Alright, so let's just erase this. Now for the blood supply. First, for the incoming blood supply, we have the anterior choroidal artery, and for the venous drainage, let me write that out, venous drainage, there's going to be one of two: lower choroidal vein or the choroid plexus vein. Alright. And for next, we'll have the regions. Now, the regions will be a bit difficult. Uh, I'll explain it as I sort of draw it. But some of these regions, um, maybe it's better if I identify the regions first. Uh, this will be the lateral region, and again, remember this is right-sided amygdala, right? So we'll have the lateral nucleus out here, the basolateral nucleus over here, down here, this area, we will have the paralaminar nucleus, we'll just say PL for that. Over here, the basomedial nucleus. Up here, the central nucleus. Medial. Over here, we'll have the ventral cortical nucleus. And over here, up here, this is for the amygdaloid piriform transition area. So I will be linking the cytoarchitectonic map that I'm using in the description below. But if you look at other researchers and regions they use, there will be distinctions. They won't use the exact regions as outlined here, but this one is a very good cytoarchitectonic map where you'll always be able to understand the researcher's nucleus and how they've subdivided it. And unfortunately, that is the case when looking at the amygdala. There's a lot of different approaches, and I might link that if anyone's interested in the description below. There's a paper talking about looking at I think 5 or 6 different studies, and it showed each one of the researchers used a different nomenclature for labeling the amygdala. But this one is fairly good, and if you look at a lot of research going on in the amygdala area, this maps pretty well, and it gives you a good idea. I should also draw out some of the outer lying areas. Down here we'll have the hippocampus. Over here we'll have the entorhinal cortex. Up here we will have the claustrum. And I think this is fairly good for mapping out. Maybe, yeah, I could put the nucleus basalis of Mevnert. Spelled like this, Mevnert. But if you look at the link in the description, it will give you a very good indication of what all these areas are. All right. So within these, these will be called the nuclei, these specific regions, but they are often grouped into clusters or complexes. So in the link that I've sent, this is the centromedial complex. This one, let me do this one in black. This one is called the basolateral complex. However, you may also see it as lateral basal complex. Although I would say this seems to be more well used, but obviously this can be a bit frustrating because the basolateral nucleus is located within the basolateral complex. So that can be, yeah. And then we have the superficial amygdala here. I'll just write superficial amygdala or superficial complex. So there is also another discrepancy even with the complexes that I've seen where some researchers group these superficial areas with the medial areas and they call it the cortical medial complex, and then they just put the central area by itself. That can happen, but I think if you have an understanding of this map, you can pretty easily distinguish what they're doing. So that's why I like this cytoarchitectonic map. Now, if we get into the neuron counts, these neuron counts are a rough estimation. I'll link below the author that I'll be using, but I've seen a couple different estimates, but it's between 12 to 15 million neurons. The lateral section has about 4 million neurons. The basal section, and again, they use basal here. I assume they're meaning lateral basal, but we can, yeah. Basal, they might even mean basolateral and basalmedial, but it's about 3.24. So you can see that this basolateral complex is quite large compared to, if you just subtract those, it's quite large in terms of total neuron count. And these accessory basal, let me just write down accessory basal areas have about 1.28 million. So again, yeah, it's quite large. Central, only about 0.36, they estimated. And then they just put for the remaining areas, 3.33. Now, as a small note, we can also say types of cells. There's 3 types of cells in the amygdaloid complex. There's glial, neurons, and endothelial. Oh, let me just circle those. And if we were to look at the basolateral section, so we do BL, the author gives an example of about 70% pyramidal neurons and 30% interneurons. Okay, and now all we have left to talk about are the pathways. So just give me a second to erase all of this. All right. And so we will be dividing these into efferent and afferent pathways. Again, I probably should have written afferent first, but it's okay. This will be the output. These will be input pathways. So for efferent pathways, we have the amygdala-fugal. Amygdala-fugal pathway. You'll see this often. It's a major pathway. Stria terminalis. The anterior commissure. Now you may not see this one as much. It's much less well understood. And you might see these grouped as 3 major efferent pathways. But there's also several direct paths. We have hippocampus, or maybe better to say direct connections, the entorhinal cortex, and the thalamus. All right, and we can just write here direct connections. Okay, and if we go to the afferent, you'll notice there's amygdala-fugal and stria terminalis. So we'll just write amyg and stria. So these are bidirectional pathways. For the afferent connections, we'll also have the olfactory stria and temporal lobe structures that will be giving input. We'll talk about these things more in depth in the second video because these pathways are very important for understanding the physiology of the amygdaloid complex and the different processes that happen within. But thanks for watching, and I hope you enjoyed the content. Thank you.